Thank you, Chantal, and thank you very much to ILSI for inviting me to your meeting. It's my real pleasure to talk to you about something that is my most favorite topic. I have no conflicts to declare. John mentioned the microbiome field is moving very rapidly. We've gone over a 10-year period, we've gone from culturing microbes to identifying them according to their DNA sequence. And in doing so, we have discovered that we have more friends than we think, especially in the gut. And these friends we acquire during our infancy. And the moment we are born, there are many opportunities in terms of environmental exposures for us to accept and delete friends. So the purpose of my session is to tell you about the development or maturation of the gut microbiome during infancy, how it's affected by common early life exposures. And in doing so, I will tell you a little bit about my microbiome research program and the child birth cohort. Just Taxonomy 101, I'm going to be presenting results mostly at the phylum level, but also at the family level in terms of microbial nomenclature. The childbirth cohort, which you may or may not be familiar with, is a pregnancy cohort following 3,600 mothers, infants, full-term infants that have been recruited at 4 sites in Canada, Vancouver, Edmonton, Winnipeg, and Toronto. And so that's my map of Canada. And I originate from the second province, from the western coast. That star is Edmonton. And I'm not sure whether Ireland is the size of Alberta or not, but just to give you an indication of the dimensions of Canada. The childbirth cohort was constructed to study the most likely or early life exposures that are associated with chronic disease, allergy, asthma, overweight, and then as our children are getting older, and we have just finished the recruitment at age 5, we're starting to look at other types of health and mental health conditions. You'll notice that during the process from the initial recruitment to the follow-up, the child study has collected thousands and thousands of samples, and part of these sample collection were fecal samples that were collected at 3 months and 1 year of age. And this is where my research program comes in. I can't believe it's already 7 years ago. Was funded by CIHR with a team grant to enhance capacity for microbiome research in Canada, using samples from the childbirth cohort, sequencing them, and then linking them with metadata from the childbirth cohort to study determinants of disease, but also to assess the myriad of early life exposures that can affect this development of the microbiome. I'm going to start from the beginning and tell you a story of how the gut microbiome develops, and then discuss particular examples of deviations to this development. So let's start with what we would expect normally. This paper is already 3 years old. It's from my colleague Maria Gelmand, and it shows simply the different types of microbes at the phylum level and their course in terms of prevalence. I mean, the relative media abundance you can interpret as prevalence, change over time from birth to 2 years of age. You'll notice that in the pink, the proteobacteria decline, and then the Firmicutes and the Bacteroides expand in abundance. The Firmicutes, it's a huge group of microbes. The ones that you may be familiar with are the lactic acid bacteria, and they diminish as the lactospiraceae and other butyrate producers increase. So this is a pattern that one observes. This was a small N study. In our 1,000 infants, and this is from the child cohort, we see a similar type of pattern. In the pink, we see the diminishment of proteobacteria over time and the enhancement of the Firmicutes in light blue and the Bacteroides in dark blue. This is another way of showing how gut microbiota mature over time in infancy. It's a cluster plot. Each cluster represents an infant, and it's a representation of their community, microbial community. You'll notice that the 3-month and the 12-month clusters separate, and they're clustered together, indicating that the 3-month microbiota is different in composition than the 1-year microbiota. This is another way of showing how composition differs over infant age, and here we have some data from the child cohort on meconium, and we see it clustered separately from the 3-month and again separately from the 1 year. We've known for a while, or at least the speculation has been, that immediately after birth, that it's those lactic acid bacteria, the proteobacteria, that are facultative anaerobes, and they lay the foundation of an increasingly anaerobic environment to allow for the colonization of the Bacteroides, the bifidobacteria, and the various Firmicutes. We're at the stage now where several cohorts around the world are starting to provide evidence that what you start with determines what you end up with. So for example, from the dog paper, this is the Gusto cohort in Singapore. The higher the amount of Enterobacteriaceae, and then the family level of the proteobacteria within 3 days of birth, the more likely you will have a more abundant bifidobacteria 6 months later. A nice paper by Martinal, which characterized in detail at the species level the various lactobacilli that are present soon after birth, and then you see that with most of the species, all of the species, that they increase and then they decline over time. So as I mentioned, so these are the microbes that lay the foundation of the succession and the colonization of the Firmicutes and the Bacteroides. This is a busy slide, and if you're interested in the details of it, I've listed the citation below, but the main purpose why I like to show the slide, I like to show this because it shows in the rows the common early life exposures and their impact on the individual genus, microbes at the genus level, and then the size of the dot and the intensity of the dot indicates the impact of that particular environmental exposure, and we see that breastfeeding, no surprise, is a strong determinant, birth mode, and then antibiotic use. So basically, how you were born, what you were fed, and your exposure to antibiotics shapes your microbiome, your gut microbiome as an infant. Let's just quickly talk about cesarean section birth, because at this point, we know quite a bit about it. There are about 12 cohort studies that have published on the impact of cesarean section birth on gut microbial composition. Again, I return to the figure from the Maria John Mon group, and she also reported on what happens, what does the composition look like over time in terms of cesarean section. And we see that if you focus on the dark blue, you see considerable diminishment of the Bacteroides. Again, you don't require a large N study. This was our first publication from a child cohort of 24 infants, and we see that in the orange, the Bacteroides, they are virtually absent in our cesarean section delivered infants. So we are at a stage now, as I mentioned, with 12 cohort studies that are providing evidence that one can summarize in terms of birth and postnatal environmental factors that impact on the composition of the gut microbiome. And I just wanted to say just a few words about the first chapter. I had written it with the intent as a user-friendly chapter for those individuals who are new to the microbiome field and wanting to know about its development and those birth and postnatal factors that determine it. The main message from this chapter is that each of those exposures, cesarean section birth, maternal interpartum antibiotics, length of hospitalization, infant antibiotic use, they have similar effects in terms of the microbial composition uniformly. You see a reduction in Bacteroidetes. Now we've seen some groups putting together meta-analysis to indicate that this finding. We see some diminishment of bifidobacteria, but at the same time, some increases in the staphylococci, the Clostridia, and some proteobacterial species. Emergency section is a special case. You see additional increase in abundance of the enterococci. Now, what about maternal antepartum antibiotics, which is relatively, we didn't know very much about this standard of care. Standard of care in North America that if a woman has a positive culture, vaginal culture for group B strep, she'll receive 3 to 4 doses of IV penicillin during delivery. With this standard of care in practice and the standard of care of giving a prophylactic dose of antibiotics for cesarean section, that means minimally 40% of infants are exposed to an antibiotic by the time they are born. And in some cases, these are broad spectrum antibiotics because the mother is allergic to penicillin. This is a figure from our 2016 publication, which shows the impact of different types of birth modes, but broken down into the vaginal delivery situation with and without interpartum antibiotics, the scheduled cesarean section, and then the emergency C-section, going from left to right if you can't read the x-axis labels. And what you can see in orange, and that is at the family level, the Bacteroidetes, they're considerably reduced, even with maternal interpartum antibiotics, and definitely with elective and emergency C-section. We see some microbial species that are increased, the enterococci I mentioned, and the Clostridia. You'll notice that to the furthest to the right, that is the emergency cesarean section scenario, that this composition looks quite different from your vaginally delivered antibiotic-free infant, and there are many changes. And we're thinking that there are many things going on with emergency C-section, some of them originating in utero. But a really important point to note that we published in this paper is that infants born by emergency cesarean, their mothers may have received an additional dose, a prophylactic dose of antibiotic. They themselves are at higher risk of receiving a 24 to 48-hour course of intravenous antibiotics because of concern over infection. And then their mothers who are breastfeeding are more likely to receive a course of antibiotics due to wound infection. So in this particular situation, these infants are exposed to multiple courses of antibiotics, which potentially explains the quite divergent pattern of microbial composition. Okay, what about hospitalization? North America, most infants are born in the hospital. We don't know very much about the effect of hospitalization. If you're delivered vaginally, you could be home the same day. If you're delivered by cesarean section, you may be home 3 days later. So again, from the child cohort, we've looked at the duration of hospitalization after birth, and this is just in all infants. I think the N of this is about 1,000 infants. And then we see here the Bacteroidetes are in green. They're reduced. At the same time, you see an expansion of the Enterobacteriaceae, of the proteobacteria, and also of the Lactobacilli. So I just mentioned that the length of stay will depend on how you're delivered. So this is an overall look, but really it's not that useful. One needs to then divide these infants according to whether they were delivered vaginally or by cesarean delivery, because we know the cesarean delivered infants are already staying longer in hospital. And when we do so, we see those changes. They are more statistically significant in the cesarean section delivered infants, namely that the Lactobacilli, and this is again at the family level, are increased. Just remember Lactobacilli. Those groups of microbes will come up in my subsequent slides. Okay. So what happens later on in infancy? So far, I've been presenting to you results at the age of 3 to 4 months. What happens with the gut microbiota development later on? And what happens when you just change one exposure versus changing all of the exposures that I mentioned? And so here on the left-hand side, we have a schema showing what would we expect in an infant that was delivered vaginally breastfed and did not receive antibiotics. And you have microbes that are increased in red, those that are decreased in green, and then the unchanged ones in black. And then changing just one exposure to your right, and that is not breastfeeding. We see the same figure, but you'll notice that the slope is flatter. We see a greater number in black of microbes that are unchanged, and we see fewer that are increased in red and fewer decreased. And also the type of microbes that are increased and decreased differ. We published this paper just at the end of last year, and this is a schema just to show, to summarize the findings and to convey the message that in the breastfed, vaginally delivered scenario, we have greater increases in the Villanaceae, Clostridiaceae, versus the formula-fed infants. So we have a larger proportion of those infants that have unchanging abundance of Enterobacteriaceae. So it's not just who microbes that are increased or decreased, also those ones that remain unchanged. And if you are, even if you're vaginally delivered and formula-fed, more likely that you'll have those unchanging Enterobacteriaceae. Many mothers email me concerned that they've had a cesarean section delivery and they want to know how they can modify, they can fix their infant's gut microbiome. So I'll talk a little bit about breastfeeding now. For certain, breastfeeding at 3 to 4 months age, as shown in this diagram, does have a profound influence on the microbial composition. When you're comparing not breastfed to partially breastfed or exclusively breastfed infants. Specifically, we can identify some groups of microbiota at the family level, and again, the Lactobacilli group comes out as being increased in abundance in your infants that are not breastfed or only partially breastfed. And also, in the child study, we were looking at the microbial, in addition to the microbial composition, we are also looking at the metabolite composition, and specifically because microbes produce short-chain fatty acids. In this paper, we reported on the short-chain fatty acid composition according to breastfeeding status. The formula-fed infant, all of the short-chain fatty acids are increased in abundance. This is as absolute values. But when we start to calculate relative values, we see that, as others have reported, that in your exclusively breastfed infant, that the proportion of acetate is increased. And I think this is important because several animal model papers have identified that acetate, along with some of the other short-chain fatty acids such as propionate, are associated with reduced risk of allergic disease. Getting back to breastfeeding at 3 months of age in interaction with birth mode. So this figure shows the relative abundance according to exclusively breastfed and not exclusively breastfed infants. In the orange, we see that the Bacteroideaceae are reduced. This is at 3 months of age, independent of breastfeeding status. So we can say, as others have reported, that cesarean section leaves an imprint that is independent of breastfeeding. However, in our 2016 paper, we found that early breastfeeding changed the abundance of the Bacteroideaceae 1 year later. So we see this imprint at 3 months of age, but in those infants that are exclusively breastfed, again, this is at 3 months of age, not at 1 year of age, that early breastfeeding, 1 year later, there was no difference in abundance of the Bacteroideaceae across the delivery groups. This might indicate that breastfeeding could restore eventually some of the changes that were induced by cesarean section. But I do want to emphasize that at 3 months of age, you do see that initial imprint. I've talked to you mostly about birth and postnatal factors, but we're now very interested in looking at what happens during pregnancy in terms of maternal health status and the postnatal development of the infant gut microbiome. You may not believe in the hypothesis of the internal microbial transfer from the gut to breast milk or other parts, but definitely maternal metabolites that are produced in the gut do make their way to breast milk, and they do cross the placenta. So being very much interested in this area, and the 4 minutes or 5 minutes that I have left, I want to talk about 3 maternal health conditions and their impact on the postnatal development of infant gut microbiota, and that is maternal overweight, maternal stress, just to follow up on John's, some of his results, and then more recently, our paper on maternal asthma. So this will be quick, unfortunately, but I just wanted to give you a sense of where the field is going. This paper will soon be published. This is an example of a mediation analysis. I just want to quickly add that in terms of the conceptual framework that Gary had shown you, that there is a subclass of observational studies in terms of statistical analysis, I think, that brings us closer to causation. So it's an analytical step that one can take between the report of an association to planning your animal model. And this is, some of you maybe are familiar with this technique, it's the mediation analysis. In this particular paper, we wanted to know whether gut microbiota were the intermediates, or the gut microbial composition was the intermediate between associations of maternal overweight and child overweight. That association is very well documented. We speculate it is due to some change in gut microbiota. In this particular paper, we tested this using mediation analysis. But we didn't test the gut microbial composition on its own. We tested it sequentially, knowing that the microbial composition that you're born with depends on how you were delivered. So if you're starting off with something different, according to cesarean section of vaginal delivery, there should be some sort of interaction in terms of microbial composition and the pathway that is taken from the maternal overweight to the child overweight scenario. And indeed, we found that the maternal overweight and child overweight association was mediated by the abundance of Lactobacilli, again, the Lactobacilli, but it was sequentially mediated with the birth mode variable. And we also found that depending on whether the delivery was vaginal or by cesarean, at the genus level, the different Lactobacilli were involved. So just look out for this paper if you're interested. Now I'm changing topics and to share with you some new results on the pre and postnatal depression. This is from the child cohort, and instead of reporting on microbiota, we're now reporting on host response to microbiota. Secreted IgA is a very important component of gut microbial maturation, and I refer to the IgA molecule as involved in training the immune system to be tolerant, for example, to be tolerant to food, to be tolerant to your commensal microbes. Breast milk is the main source of IgA. In newborns, within a month, they start to produce their own. So here we reported last year that in those infants born to mothers with pre- and postnatal depressive symptoms, higher symptoms, that those infants had the lowest levels of fecal IgA. just a reminder of some of the functions of IgA, and that breast milk is a major source, but at 3 months of age, when it was measured in our study, infants were already producing their own. We know this because we also detected fecal IgA, secretory IgA, in formula-fed infants. We've also had a paper that talks about the inverse association between fecal IgA levels and Clostridium difficile, if you're interested. Now, getting back to, this was a master's thesis of my student Leanne King, showing further, stratifying the results according to breastfeeding status. This type of analysis is needed, of course, to disentangle the original association, because we know that women who have higher levels of depressive symptoms postnatally are less likely to breastfeed. So is this a breastfeeding phenomenon, or is it something in addition? And so when we stratify by breastfeeding status, so going from the left to the right, exclusively breastfed, partially breastfed, or not, we see that the statistically reduced levels are in predominantly in those women who do not breastfeed, so those women who have pre- and postnatal depression and do not breastfeed. However, also in the exclusively breastfed infant, if mom has pre- and postnatal depression, you also see lower levels of IgA. Okay. Coming to the end, and our paper that came out last year, reporting on maternal asthma. Treatment for maternal asthma during pregnancy and impact on infant gut microbiota. Why would one be interested in that question? Well, it's a significant risk factor for child asthma, especially in boys. It's long assumed to be a genetic association. However, maternal asthma during pregnancy also affects birth weight. So this led me to hypothesize that if that was the case, that it might also affect infant gut microbial development. And so here we see stratified, and this also, these findings are sex-specific. So in the male, if mom had asthma symptoms during pregnancy, those males were more likely to have a lower abundance of lactobacilli. This was not observed in females. I'm showing you two figures. One figure where the maternal asthma is combined with a positive atopic sensitization, and then the other where maternal asthma is combined with overweight, quite a common comorbidity. And we see in both instances that the infant, and it is the male infant, has a reduced abundance of lactobacilli. Females responded in a different way. So in females exposed to the same, if you want to call it, environmental insult during pregnancy, there were no changes to lactobacilli. However, they responded with increased abundance of Bacteroideaceae. And it is our hypothesis that this initial response by males versus females increases the risk for early life asthma in boys, but may protect girls till maybe potentially later and during puberty, where this increased Bacteroideaceae may have a role. Okay. And I'm over time, so I'm just going to quickly mention that we have a paper on food sensitization that came out a number of years ago, and I want to thank my team. Part of it is at the University of Toronto in terms of the DNA extraction and sequencing of fecal samples. This is my team in Edmonton, and my advertisement. And then finally, just another little advertisement of the beautiful Rocky Mountains and Canmore. That's an elk, by the way. And just to mention that I'm organizing a meeting there in a few months. Thank you.